In microbiology lab, we have hundreds of different types of media that we use to isolate and identify bacteria. These media can be classified into different groups: non-selective, selective, differential, and enrichment or enhancement media. The first media that we're going to deal with is the non-selective media, which is a TSA plate incorporated with sheep blood. The non-selective medias allow most non-fastidious organisms to grow. So we are able to recover most organisms, such as gram-positive organisms and gram-negative organisms. And you can see on these two plates we have a Staph aureus and an E. coli, and they both grow very well on the non-selective medium. Next, we have selective medium, which supports the growth of one type of an organism and inhibits another type of organism. These mediums have antibiotics or dyes or alcohols in them to inhibit the growth of certain organisms. So we have a Columbia CNA agar, which has colistin and nalidixic acid incorporated, and this will allow our gram-positive organisms to grow, but will inhibit the gram-negative organisms. Next, we have another selective medium, which is our MacConkey agar. And the MacConkey agar will allow our gram-negative organisms to grow and inhibit our gram-positive organisms. And you can see that the Staph aureus on the MacConkey agar did not grow, but we do have gram-negatives of E. coli and Pseudomonas, which have grown very well. Next, we have differential agar, and our differential agars allow us to, I messed up. That was good. MacConkey is a differential agar in that it allows groups of microbes to be visually distinguished by the appearance of the colony or the surrounding media based on biochemical differences between the two groups. MacConkey agar will distinguish a lactose fermenting gram-negative bacilli from a non-lactose fermenting gram-negative bacilli. When the gram-negative bacilli ferments lactose, you will get a pink color. The organism will actually utilize the lactose, and the dye in the media will be absorbed by the bacteria, and it is a pink color. The organisms that do not utilize lactose or do not ferment the lactose will be clear. And you can see we have a Pseudomonas that is not utilizing the lactose, and the colonies are clear. HE agar is a differential medium in that it will distinguish our non-pathogenic enterics by turning a salmon or orangey color, and it will help us find a salmonella or shigella that is in the stool mixed with the non-enteric pathogens, in that it will be a clear or a green colony if it is a salmonella or a shigella. The HE agar also has a component that will show us when an organism produces H2S, and that organism will also have black precipitants in the clear colony, such as salmonella. Sorbitol MacConkey is also a differential medium, and it helps us to identify E. coli 0157H7. On the MacConkey sorbitol agar, if the E. coli utilizes sorbitol, it will turn a pink color. If it is a non-sorbitol fermenting organism, such as the E. coli 0157H7, it will be a clear colony. The next category of media is our enrichment media, and chocolate agar is considered an enrichment media. Chocolate agar is red blood cells that have been lysed and then incorporated into the molten tryptase soy agar, and will then have X and V factors, will help the fastidious organisms to grow. And as you can see, we have got a Staph aureus and an E. coli, so it does not inhibit any growth. It allows and enhances growth of most organisms.